Hey everybody, today we're going to talk about genetics and heredity. We've already talked about meiosis, the process that cells go through in order to create gametes or eggs and sperm. Now, when a parent organism makes egg or sperm, it's going to split the DNA in half so that each egg and sperm gets one copy of every gene or one set of DNA. And when that egg and sperm come together, they're each going to donate a set of DNA so that the offspring has two sets of DNA. And so we're going to look at how those genes are passed today and how they're read to tell you what that organism is going to look like or function like. Here you can see a pea plant, and peas were very important in our understanding of genetics. Remember that flowers have male and female parts, and a pea plant has these two petals here, which are going to cover up those male and female parts so that they're not exposed to the outside world and pollination isn't going to happen. This is Gregor Mendel, and he's responsible for what we now know about genetics. He was a very educated man, and he lived in the 19th century. And at this time, educated people would often enter religious orders. So in the mid 1800s, Gregor Mendel moved from Austria to what is now the Czech Republic, and he lived in this monastery. This is called St. Thomas's Abbey. Monks had three parts to their daily lives: they read, they worked, and they prayed. And that was the life of a monk. At this time, religion and science were also one and the same. So a lot of our early scientific principles were discovered by people working in monasteries. But over time, religion, science, as of every other job, they start to separate and they start to specialize. So Gregor Mendel's job in the monastery was to be a gardener. He grew food for other monks, and he studied pea plants while he did this. He grew tens of thousands of pea plants, kept very meticulous records on their traits and on their growth. And all of his records went unnoticed throughout his life. So when he died, it took about 100 years for someone to read his work and think, oh man, this guy was on to something. And then his ideas became what we now know as modern genetics. So we said that Gregor Mendel studied peas. He looked at tens of thousands of them, and he looked at peas because they self-pollinate. They have that set of petals that's going to cover up their male and female parts so that the sperm from one flower can only fertilize the eggs from the same flower. And this means they're going to reproduce to make true breeding organisms. True breeding organisms have homozygous genotypes. That means that both copies for a particular gene in that organism are going to be the same form. And it's like that for every type of gene they have. Both copies for that trait are the same. They're homozygous. So that means every time that parent reproduces, it's going to donate the same gene form for a particular gene because they don't have but one type of gene form. So he noticed that when peas with different traits were bred, all plants in the first generation would have the same traits, and it would be a trait from one particular type of plant. So he looked at things like seeds, if they were round or wrinkled, if they were yellow or green. He looked at flower color, whether the pea was white or purple. He looked at pods, whether they were full and smooth or whether they were constricted and tightly packed up against the seeds in the pod. He looked at color, if they were yellow or green, and he looked at stems, whether this flower bloomed on the sides of the stem or whether the flowers only came out at the end of the stem. So axial means they come out on the sides. Terminal means you only have flowers at the end of the vine. And he also looked at whether or not these stems were long or short. And when he would breed plants that were long with plants that were short, he would notice that the first generation of these plants all had the same traits. So this led him to thinking something was being passed on from parent to offspring. So this is where the idea of heredity was born, the passing of traits from parent to offspring. And most single-celled eukaryotic organisms, that U means true and karyo means nucleus. So these are unicellular organisms with a true nucleus. They're going to inherit their genes asexually through mitosis, making an identical copy of those cells. Prokaryotes, pro means before and karyo means nucleus. So these evolved before there was a nucleus and they don't have one. So they're going to inherit their genes asexually through a process called binary fission. Remember, they don't have a nucleus, and mitosis is division of the nucleus. So prokaryotes can't do mitosis because they don't have a nucleus. The chromosomes between eukaryotes and prokaryotes are going to be a little bit different. Eukaryotes have these rod-shaped or linear chromosomes that we find in the nucleus. Prokaryotes have these circular chromosomes. They connect end to end, and they're found in the cytoplasm, again, because they don't have a nucleus. So these genes are going to be passed in sexually reproducing organisms in homologous pairs. Remember, with sexual reproduction, there are two parents, and each parent passes on a set of chromosomes. So these homologous pairs, you get one of them from each parent. Mom gives you a chromosome, and dad gives you a chromosome. They have the same types of genes on them, but they might not have the same forms of the gene. So, for example, dad could give you brown hair while mom gives you blonde hair. It's still hair color. It's the same type of gene, but it might not be the same form of the gene found on a homologous pair of chromosomes. So, when you hear me say allele, this is just the form of a gene. Brown hair would be an allele, blonde hair would be an allele, red hair, black hair, these are all alleles. It's the same type of gene, but there's different forms of it. A genotype is all of the alleles in an organism for a particular trait. And we have two of every gene, so we have two alleles for every gene. And you can either be homozygous and have the same alleles for that gene, or you can be heterozygous and your alleles can be different. Homo means same, hetero means different. So, for an example, you could have both parents gave you blonde hair. That would be a homozygous genotype because your alleles for hair color are the same. If dad gave you blonde and mom gave you brown, that would be heterozygous because your alleles for hair color would be different. Genotypes result in a phenotype. This is the outward appearance of a gene. So what color you actually see. Dad gave you brown hair, mom gave you blonde hair, you might have brown hair. So the phenotype would be the brown hair. The genotype, you would be heterozygous for hair color. So here we can see a homozygous fly. It's got two genes that are the same. They're represented with capital letters. And this is going to give us normal wings as our phenotypes, what it's going to look like. Here you can see flies that have been given different genes, a big W and a little w. So one gene for normal and one gene for wrinkled wings. And you can see here it's been reversed. Either way, you have the copy of that normal trait. So the phenotype that you see is normal wings. When you have two alleles for wrinkled wings, this is when the organism's phenotype is going to code for being wrinkled, and that's what you'll see. So phenotype is the outward appearance. Genotype is the combination of alleles you've got. We talked about the law of independent assortment when we talked about meiosis. Meiosis is the process that cells go through in order to create gametes or eggs and sperm. And when egg and sperm are created, the gene that goes into one of those eggs has nothing to do with the other genes that is going to go into the same egg. So, for example, the gene that goes into the egg for hair color has absolutely nothing to do with the gene that goes into the egg for eye color. And neither of those are going to have anything to do with whether or not that gamete has a code for rolling its tongue, doing this. Okay? So genes separate from one another randomly and independently, and they do not influence each other in how they're going to be put into that gamete. Now, Mendel was the first person who observed this, and he didn't know anything about chromosomes and how genes are arranged on a chromosome. So there are many genes on the same chromosome, and it may seem that those genes are going to be linked together, always passed together. But remember, with crossing over, we can still separate those genes and put them on different chromosomes and put them into different gametes. And so independent assortment can work using crossing over. And Mendel observed this with his peas. He noticed that the gene for whether or not this pod was constricted or smooth had nothing to do with whether or not this pod was yellow or green. So you can get yellow smooth pods and you can get yellowed wrinkled pods. You can get green wrinkled pods or you can get green smooth pods. But those genes are not linked together. Just because you have a yellow pod doesn't mean it's going to have to be constricted. So big pieces of DNA are called chromosomes, and little pieces of DNA on those chromosomes that code for a certain protein are called genes. So proteins are coded for by DNA. And these proteins that an organism makes are going to control its traits, what it appears like, what it functions like. And this process of going from DNA to protein is the central dogma of molecular biology. So we start with DNA, it's transcribed to mRNA, which is translated to protein. That's the central dogma. And the accumulation of this protein is going to result in a trait, what an organism looks like. Now, chromosomes in most eukaryotes, anything that reproduces sexually, are going to be found in homologous pairs. So you have a chromosome and its matching chromosome. They have the same types of genes, but they might have different alleles or different forms of those genes. And you can see here a chromosome and its exact copy. These are sister chromatids. And here you can see the homologue of this chromosome. So they have the same types of genes in the same location, but they might have different forms. So remember we talked about homozygous and heterozygous. Hetero means different. So these genes are heterozygous. They have different forms. These genes are the same. They're represented in the same colors. So these are homozygous. Homozygous. Homo, hetero, they're different. Hetero, homozygous, heterozygous, heterozygous, homozygous, and this last one would be heterozygous. Most traits are controlled by multiple genes. There's not just one spot in your chromosomes that's going to control for that trait. There's many different places in your chromosome where you have genes that control for that trait. So for example, hair color, it's not just that mom gives you one gene for hair and dad gives you one gene for hair and you have either blonde or black. You actually have about 6 places in your chromosomes that code for your hair color. And mom and dad each give you 6 genes for coding for your hair color. So that's why you get this wide variety of hair tones. Most of these traits are going to be additive. So the more alleles you have for a particular trait, then the stronger that trait is going to appear to be. So if you've got more brown alleles in your hair color, then your hair is going to appear more brown. Now, some alleles are coded for in one place in your DNA. Some genes are coded for in one place in your DNA. And there are some alleles that can mask the appearance of another. So we call those types of traits dominant or recessive. A dominant allele would be the ability to roll your tongue, to do that. Not being able to roll your tongue is recessive. So as long as mom or dad gave you one gene that says you can roll your tongue, then you can roll it. If neither of them did, then you cannot roll it. And so these traits are only coded for in one place in your chromosomes. We're going to talk about these more tomorrow. The environment does play a big role in gene expression. Your skin tone, for example, if you go outside and you spend time in the sun, the environment can trigger the production of a protein called melanin, and your skin's going to get darker. You're going to get a tan. So that's an example of how environment can play a role in your gene expression. You can see here a map of different skin tones and where you're most likely to find them. And you can see skin tones generally get lighter as you go away from the equator where the sun is less intense. Over here, we have hydrangeas. These are snowball bushes. And you have different colors of hydrangeas based on the soil that they grow in and the pH of that soil, how acidic or basic it is. Acidic soils are going to give you this blue color in hydrangeas, while more neutral soils are going to give you this pink color. And you can have different shades with pHs that are between 4.5 and 7. So you can actually change the color of these flowers from year to year by changing how acidic or basic your soil is. So now we're going to look at some genetic crosses. This Punnett square, it's already been filled out. You can see another one there on the board behind me that is blank. These are tools that scientists use to determine the probability of a genetic cross. So what are the chances that I will find this particular trait in my offspring given the traits of my 2 parents? So these are mostly going to be used for traits that involve one gene. Now you might have noticed earlier, you can have really big Punnett squares. We're not worried about those in this class. We're going to keep it simple, and we're only going to look at crosses with one gene. And there are mathematical tricks that you can use to figure out your chances of getting a combination of genes, but we're not going to worry about that for this class. This Punnett square, we divide it into 4 sections, and we use letters to represent our genes. So this is a homozygous dominant genotype. It's got 2 of the same letters, and they're both big to represent dominant. This is a heterozygous genotype. Both of the letters are different cases, so we have different genes. And this is a homozygous recessive genotype. Both letters are the same, and they're both little to represent recessive. We're going to go into dominant and recessive more tomorrow. Now, we're going to write our parent alleles across the top and across the side. And here's the kicker. You have to keep the parent alleles together. So I can put dad's alleles here, or I can put dad's alleles here. Okay, I can switch around where I put them, but I can't put one of dad's alleles here and the other over here. I can't cut my corner. Okay, but you can switch around where you put mom and dad's genes. That's fine. So these represent my gametes. This is my parent organism. It had both of these genes. And during meiosis, it's going to split those genes into two different sperm. This is my mother plant. It had both of these genes in the organism. And during meiosis, it's going to split those genes into different eggs. Now we're going to cross these alleles to find our genetic cross, see what our offspring are going to look like. And to do that, we're going to bring the things that are on the top down. So there's my big B, there's my big B. And we're going to bring this one down, little b and little b. And we're going to bring these things across. So there's my other big B, here's my little b, and here's my other little b. So this represents fertilization. These sperm and eggs are coming together. And this cross, this final outcome, represents what your zygote is going to look like. Now we can express these in ratios or percentages. I could say I have 25% chance of getting this homozygous recessive genotype. I have a 50% chance of getting this heterozygous genotype. I have a 75% chance of getting purple flowers. I have 3 purple flowers to 1. These are ways that you can represent your crosses. We'll look more at that here in a second. These are your types of inheritance patterns that we're going to go over. We're going to focus on these top 3. We are going to do some genetic crosses with multiple alleles when we look at blood types, but mostly we're going to be focused on these top 3. These are the rules for solving a genetics problem. Now, I haven't asked you to list these in your note guide, but you might want to copy and paste them there so that you can use them. First, you need to know the type of cross, and then you need to determine the genotype of the parents. Once that's determined, you can fill out your Punnett square. You can do your cross and determine what your offspring's genotypes are going to be. Then you can analyze the Punnett square by determining what those offspring are going to look like, the phenotype. But don't forget to answer the question. I can't tell you how often students will do this beautiful Punnett square and they'll come out with exactly what they need, but they never actually answer the question, and so they don't get the question correct. Genotypes are the complete set of alleles you have for a gene, and we have 2 alleles for every gene. So our genotype is going to be represented with 2 letters. Homozygous genotypes, the letters are the same or their cases are the same. And heterozygous genotypes, you're going to have different alleles, and so you'll use different letters or different cases. Phenotypes are how that trait is going to show up, and that's how we analyze these letters. So we're going to look at some of this. So you can analyze your genetic cross using a Punnett square, and you can represent your analysis with a ratio or a percentage. A ratio is just a proportion of the individuals that have the trait that you're looking for. So for example, if I do a phenotypic ratio of this cross, phenotype is what it looks like. So I have 3 brown eyes to 1 blue eye, and that would be my phenotypic ratio, 3 brown to 1 blue. I can also look at the genotypic ratio. I have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So my ratio is 1 to 2 to 1 here, okay? And these don't have to be the same, as you can see, but with what we're doing today, they will be. When traits are inherited and they are incompletely dominant, neither trait is going to dominate the other, and you'll get a blended result. So red and white make pink, for example. So incomplete dominance, you're always going to have 3 phenotypes, okay? 3 ways that this organism can look: red, white, or pink. But you're only going to have 2 alleles, an allele for red and an allele for white. Now, I really want you to use different letters when using incomplete dominance. So pretend all these little R's are W's. Now, to get a pink flower, I have to have 1 gene from both of my pure homozygous flowers. So we're going to cross a red and white snapdragon, and that will be our P generation, our parent generation. And we're going to get an F1 generation from this. So here I have my Punnett square for crossing my red and white snapdragons. Now remember, to get red, I need 2 genes for red to get that pure red color. I need 2 genes for white to get that pure white color. And to get pink, I have a blend of the red and the white. So I don't have a pink allele, I have a combination of red and white to get pink. Now, if I'm crossing a red snapdragon, I'm going to have my 2 red alleles. They go on the same side of the Punnett square. and I have my two white alleles from the other flower, and they go on the same side of the Punnett square. When I cross, I bring everything from the top down, okay? So my R's came down, and I'm going to bring everything from the side across. So everything's also going to get a W here. And that means that every single one of these plants are going to be pink. So when you are doing your cross on your note sheet, that's the answer to your question. All of them, 100%, will be pink. Okay? And this is our F1 generation, our first round of fertilization. So we had our parents, they fertilized one another, and now we have our F1. So we got the results of our F1 generation. All of those organisms were pink. And now we're going to cross two of those organisms from our F1 generation to get an F2 generation, the second round of fertilization. So I'm going to put my RW here in different gametes. Red went to one gamete, white went to the other, and same for my other flower. And I kept those alleles from one plant on one side of the Punnett square. So remember, everything from the top is going to come down, and everything from the side is going to come across. And I like to be consistent in which gene I list first. It really doesn't matter if I put RW or WR, I still have pink. Okay, I still have a heterozygous genotype. So now we can go through and analyze these. And in this box, I have a red flower. In this box, I have a pink flower. In this box, I have a pink flower. And in this box, I have a white flower. So now I can look at my ratio, and I have one red to two pink to one white. And that would be my phenotypic ratio. It's what it looks like. I can also say that I have one homozygous red. I have two heterozygous, and I have one homozygous white, and that would be my genotypic ratio. This is another way to look at this. You have your red flower, you have your white flower, and these red alleles are going to be put into different gametes, and so are these white alleles. So remember, though, I got 4 different boxes I need to put these in. So my red allele is going to go into every box. My white allele would go also into every box. And I end up with a combination of red and white alleles in every flower, because there's really no other way to do that. These are only going to donate red. These are only going to donate white. I can also look at my F1 cross. So I have 2 pink flowers with a red and a white allele in each of them. And this time I can partner red with red, or red with white, or white with red, and white with white. And so I end up getting a variety of my different phenotypes in my offspring. So basically you're using the FOIL property in math, if you remember that: the first, the outer, the inner, and the last. Codominance is like incomplete dominance, but instead of getting a blend of 2 traits like we got with incomplete dominance, we're going to see a separate showing of the traits, and it's going to be fairly equal. So here I have a flower with red and white genes, but it's not going to blend together to give me pink. Parts of the flower are going to be red and parts of the flower are going to be white. So that's why it's codominant and not incomplete dominance. Here we have a chicken. Part of it is white and parts of it are black. It's got genes for black and white, but those genes didn't mix to give you a gray chicken. They mixed to give you a spotted chicken. Now, we still have 3 phenotypes: red, white, and checkered, or black, white, and checkered with our chicken. So we're still going to use 2 letters to represent those 3 phenotypes. So we're going to practice. We're going to cross a black and white chicken, and then we're going to cross a black chicken with a black and white spotted chicken. So here's my Punnett square. Now remember, I can have 2 alleles for black. That's going to give me a black chicken. I can have 2 alleles for white, and that's going to give me a white chicken. To get my spotted chicken, I have to mix black with white, and that gives me my spotted or my checkered chicken. Now, in your problem, you've been asked to cross a black with a white chicken. So I'm going to put my alleles from the black chicken together on the top, and I'm going to put my alleles for the white chicken together on the side. Everything that's on the top is going to come down, and everything that's on the side is going to come across. And again, it doesn't matter which order you put these letters in, but I do like to be consistent and keep one certain gene first. But all of these chickens are going to be spotted chickens. every single one of them. So your ratio for this, if you needed to put it, would be 1 to 0. For every one black and white chicken you have, you've got nothing else. Okay, or 4 to 0. For every 4 black and white chickens, you have nothing else. In your next problem on your note sheet, you've been asked to cross a black chicken with a black and white spotted chicken. So I'm putting the alleles of my parents here on the side. Now I'm going to cross them to get my offspring. So we got a black chicken there and here, and we have a spotted chicken here and here. Okay, so when I go through, I want to know that these are black and these are checkered or spotted. We'll say spotted. And when I want to do a ratio for this, I can notice that if I look at my phenotypic ratio, then I have 2 black to 2 spotted. I can reduce that to 1 to 1. It's like a fraction. 2 over 2 can be reduced 1 over 1. So I can say I have 1 black to 1 spotted. Okay. When it comes to my genotypic ratio, I got to look at the letters, the actual genes. I have 2 chickens with a homozygous black genotype to 2 chickens with a heterozygous genotype. I could again reduce this 2 over 2 to 1 to 1. And I'd recommend always reducing, but I have seen it on the EOC both ways. So that's a good little overview of genetics. You've got some practice problems in labeling some different types of genotypes, determining what's a genotype and what's a phenotype, and then some different crosses. So please do those, submit that for your assignment, and please let me know if you have any questions or if you're confused at all. We'll move on to new types of genetics tomorrow.